Okay, now that leads us to the last thing here, splicing. RNA splicing is critically important, and we're going to see it has a lot of different functions, a lot of complexity about splicing. We recall that a gene or the messenger RNA transcript our gene or messenger, pre-messenger RNA, you have exon sequences. Those actually provide the instructions for making, for what amino acid join together and what part, what order to make a protein, and intron sequences that don't have those instructions. Once again, this is obviously continuous length of nucleotides and just drawing boxes to represent certain key sequences. Now, during splicing, the crude idea, is where we get rid of the exons, or rather get rid of the introns, and then glue the exons together. It's kind of like taking a recording on network TV and basically editing out all the commercials. So you have a continuous storyline instead of a whole bunch of stuff selling you cars, insurance, and God only knows what else, right? Has nothing to do with the storyline. Okay, so that's the kind of thing that we have here, this process of splicing. Now what you have to do, think of this, you have to do several important things when you do splicing. One of them, you have to be able to recognize what is the boundary between an intron and an exon. Now in these drawings, of course, I do them as boxes, but in real life, it's just nucleotide sequence. That's telling us that somehow there must be some features at the boundary between an exon and an intron that is universally recognized. So we have to be able to recognize it. Okay, that's one thing. Now imagine what would happen if that recognition process was really lousy. You could end up, instead of splicing all the exons together, you could end up cutting here and throwing in some intron sequence into your final messenger RNA that you don't want. That could screw up the entire protein, because now you're putting amino acids you don't want in there. You may have done what they call a frameshift, meaning you got things out of register, and that could screw everything up. Or suppose you accidentally cut into an exon. Well, if you do that, you're going to have part of an exon instead of the whole thing. Now, as we're going to see, in some cases that actually does happen, but that's a deliberate thing in order to modify the final messenger RNA. In one cell, you make one protein. In another cell, you make a different protein, different version of that protein. So we'll see that later. That sometimes actually happens, but that's an intentional type of thing. But, so definitely, in general, you're not going to want to add intron sequences or remove exon sequences. You have to recognize those boundaries, and you have to recognize them very accurately. Okay, second thing, when you're doing splicing, what you have to do is you have to hold the exons together, right? You don't want them falling off. You have to cut right at the boundaries. And of course, when we end up cutting an RNA molecule, that's going to be a hydrolysis reaction, where you're breaking a covalent bond from one nucleotide to the next. Now, those are exergonic reactions, so it doesn't take energy to do so. You have to catalyze it. So, and you have to have an enzyme that recognizes those intron-exon junctions and cuts right there. Okay, at the same time, you can't let the exons drift away from each other. You have to hold the exons together while you're slicing out the intron sequences. Now, the third thing that we have to do, we then have to join the exons together. Now, what that means is you're going to make a covalent bond from the nucleotide of one exon to a nucleotide of the next exon. That's a condensation reaction, an endergonic reaction. Okay, so the process of splicing is actually fairly complex. You have to recognize boundaries, cut specifically at those boundaries, that's one type of enzymatic activity, and then do a different type of enzymatic activity to join the exons together. So that's a pretty big bill of goods to do this kind of process. So not surprisingly, what does splicing is a very large complex of proteins and RNAs that's called the spliceosome. Now, by the way, although we're focusing on what happens with polymerase II precursors, in other words, protein-coding RNAs, there are other RNAs where you have to cut, and in some cases, cut and splice things together. So there are going to be different types of spliceosomes that can be used for other types of RNAs that need processing as well. But we're going to focus on the one that is perhaps the most important, certainly the best study, the one that does the polymerase II things. Now, needless to say, spliceosome is going to have to be pretty complex to do all these kinds of things in one single complex of molecules. So spliceosome is gigantic. Molecular weights are probably running in the millions. It's huge. And it's made of a whole bunch of different protein and RNA subunits. It's got several different kinds of things in it. First of all, it has these little RNA and protein complexes that are called what are called the small nuclear RNPs, or often abbreviated as SNRPs. Okay? Now, what does it mean? RNP means ribonucleoprotein. What that means is RNA protein complexes. So it tells you these small nuclear RNPs, these SNRPs are themselves complexes of RNA and protein. And what they typically are is you have a single small nuclear RNA, and then you have proteins associated with it. In fact, it turns out the major splicing ones have a set of common proteins. There are some others that don't. So a common protein core that associates, all these proteins in there are very basic, in other words, highly positively charged, so they stick to nucleic acids really well. So we have these common proteins that form like a protein core here, and then each SNRP also has unique individual ones. And the spliceosome consists of several different SNRPs. Now, they assemble together at various times. The spliceosome is not a static structure. You have parts of it assembling together to do one thing, then things pop off, and then new things come in. And so more on that when we look at some more of the details in splicing. But we have several of these things. Now, for some reason, I don't know what it was, but the individual small nuclear RNAs, they were named U1, U2, U3, and so on. Okay, and the SNRPs that contain these, each SNRP has one of these small nuclear RNAs, the common set of proteins, at least most of them do, and then several unique proteins. Okay, so we have one called U1, U2, U4, U5, and U6. This is what makes the spliceosome the major components of the spliceosome. Now you notice there's no U3 there. Reason why? U3 is involved in spliceosomes that are going to process ribosomal RNAs. So we leave them out of the ones for the messenger RNAs. Okay, so this complex, so you have that. You have these 5 separate things. And then you have a bunch of additional proteins that are called splicing factors. And there are general splicing factors that are required in all splicing reactions. And then there are specific splicing factors. And those specific splicing factors are involved in the splicing of only certain RNA precursors and not in others. Now, in some senses, this reminds us of the basal complex, a huge complex of multiple proteins. And the basal complex, you have these general or basal transcription factors, but then you're going to have specific transcription factors that will enhance or reduce the transcription of a particular gene. We're seeing a similar kind of thing here. You have your basic spliceosome, the 5 different SNRPs and some general splicing factors. That's the minimum you need for splicing. But then we're going to have specific splicing factors. And those will either enhance or inhibit splicing of particular RNAs. So spliceosomes are large, complex, and very dynamic structures. Like I said, we'll take a little look at this later on about sort of the assembly and the process of splicing itself, and then we'll see how we get specific splicing. Okay, well, so it's a fairly large complex here. Okay, now, interesting thing is there's been a lot of studies. The RNA splicing was first discovered in about the early 1980s, and people have noticed when they looked at using RNA probes, when they looked at messenger RNAs and they could use actually under the level of the electron microscope, they could see something like here's a messenger RNA and here's our probe. You can actually see it in some cases. You can label these things with little tiny balls of gold, and they can actually see it under the electron microscope. When they looked at messenger RNA, so it's nice one-for-one correspondence. When they looked at nuclear RNA, they could see something like this. They had these loops that were not in the original messenger RNA and the final messenger RNA. So it looked like these loops were cut out in the nucleus. And when that was first discovered independently by 2 labs, one at Cold Spring Harbor and one at, I think he's at MIT. And they realized that it must mean that certain portions of messenger RNA precursors are chopped out and thrown out in the nucleus. And the final messenger RNA, you have these sections spliced together and other sections discarded. Okay, when those discoveries were made independently by 2 major labs, both of those labs ended up getting a trip for Stockholm for that. Yes?
The linear one is the final, correct?
Yeah.
So are they thinking that when you cut it up, that it just has a curve, or that the curved portions are the introns?
Oh, well, we know now the curved portions are the introns. But the first discovery, and this is where it led to the idea of splicing. They look at the mRNA, you see this nice one-for-one correspondence, right? They look at the nuclear RNA, you see this probe bridging an area that's not in the final messenger RNA, this loop. That meant, since you don't find the final messenger RNA, it meant that loop must have been torn out and thrown away. And then these sides of the loop were glued together. So that's when they got the first idea that RNAs are spliced. Portions are discarded and other portions are joined together in the nucleus. And they showed evidence for that, and they got trips to Stockholm. One little interesting sideline, though, I will have to say, this is total sideline. The 2 labs they got, there's a guy named Rich Roberts at Cold Spring Harbor. He shared a Nobel Prize with the other lab leader, Phil Sharp, and I think MIT. Okay, well, when my wife was a lab tech down at UAB for a number of years, she spent like a decade working in this one lab, and it was a husband-wife team that ran the lab. One of them worked in Rich Roberts' lab, and did a lot of the work that led to this. She did a lot of the work where she saw these loops and stuff. Her boss got the Nobel Prize, she didn't. There was a lot of controversy about that. Many said that she definitely deserved to go up to Stockholm with him, but she didn't. Having personally met Rich Roberts at a conference or two, I would say the guy, put it this way, he's a British guy, and my impression of him was that he would make a vat of liquid helium look warm and fuzzy. I mean, you want to talk about Iceman. This guy was not just cold, he was cryogenic. So I could imagine I would not want to have worked under him, as brilliant as he was and stuff. But yeah, there was a lot. If you mentioned to this particular person that's at UAB the name Rich Roberts, you would definitely get a reaction. She did her postdoc under it. But anyway, yeah, she probably should have gone up there. She should have probably gone to Stockholm along with them, because she actually did the work that led to that discovery. Okay, well anyway, that was a very important thing, and then everybody was going after splicing and stuff. But now we know a lot more about it. And the spliceosome is a critical thing. It's a very complex set of enzymes. Now, an interesting thing about it, you're dealing with enzymatic reactions here. You're dealing with hydrolysis reactions. That's where you cut things. You're dealing with condensation reactions. That's where you join the exons together. Now, one thing that's kind of interesting is what part of the spliceosome does it. And it's now widely believed that the active sites of what you would say the enzymatic activity are kind of a hybrid, where in order for those enzymatic activities to function, you need both portions of those small nuclear RNAs and some of the proteins. The proteins by themselves cannot do those splicing reactions. The RNAs contribute. One time they thought the RNAs perhaps were the ones that were actually doing all the work, meaning that these small nuclear RNPs were ribosomes. Now they're leaning towards the active site being both part of the RNA and part of some of the proteins, sort of a hybrid active site. So that's an interesting thing. We're seeing at least RNAs that, once again, contribute to catalytic reactions. Although they may not be the only things that do it, the proteins probably play a role too. So that's an interesting kind of thing here. So we have a lot of complex stuff going on with these spliceosomes. Now this idea of specific transcription specific splicing factors, that gives us some insight into why we can mistranscribe a given gene, and yet it never leaves the nucleus. Because, for instance, the globin gene, apparently to splice the globin messenger RNA together, you need a specific splicing factor, or maybe more than one. If you don't have them, for some reason the spliceosome isn't able to do the job and splice those globin RNAs properly. Only the erythroblast, the red blood cell precursors, have those splicing factors. So with red blood cell precursors, the globin RNA gets properly processed, properly spliced, and then it gets shipped out as a nice, happy, mature messenger RNA to the cytoplasm. Therefore, the globin genes are expressed. Other cell types don't have those splicing factors. So the globin pre-messenger RNA, the nuclear RNA, is hanging around there waiting to be spliced, and sorry folks, we don't do your kind around here. They're lacking the splicing factors. Spliceosomes never do their work, so it's hanging around complete with intron sequences and everything, and eventually the nucleases recognize and say, we don't want you here, and attack them and destroy it. So, a lot of times what it must mean is that cells have numerous specific splicing factors that are cell-specific. That's pretty cool. And that is what allows us to make sure that mistranscribed genes never get processed and make it out into the cytoplasm. So that's an important thing about splicing. Okay, a few more things about splicing. We don't have the time to go through the process of splicing. We'll do that on Tuesday. But anyway, a few more things about splicing. We're going to go into this in more detail, but the idea of alternate splicing. And just a quick and dirty about this. Okay, let's make a gene here. Or rather, we'll make a messenger RNA, or a precursor to a messenger RNA. And these are going to represent our exons. Depending on what type of specific splicing factors you have, you can choose to discard certain exons and keep others. Or maybe not discard any of them at all. So you can select same precursor, same nuclear RNA. In one cell, let's suppose you splice all 4 of the exons together. Here's our mRNAs. That's great. You made one type of protein. And another cell type, let's suppose we only do 1, 3, and 4, and we eliminate exon 2. That's going to give us a different type of protein. It may be related, but it may well have different functions or do things differently. In another cell, let's suppose we have 2, 3, and 4. Still a different one. And maybe a different one in a different cell type. 1, 3, and 4. Oops, I already did that. How about 1, 2, and 4? Okay. So we can see, and there's other combinations too, we can see that the same precursor nuclear RNA can lead to several distinctly different messenger RNAs. Now this can be one cell type, another cell type, another cell type, another cell type. So that way we get 4 cell-specific versions, and there's more possibilities than I just drew, in 4 different cell types. Great. Or at one point in development, we do this. At another point in development, we do this. Later on in development, we do this. So we could change the splicing pattern as time proceeds and the cell differentiates. And the advantage of this type of alternate splicing is the old saying, and this was like, you know, this was holy gospel when I was in college. 1 gene, 1 protein. Anybody remember hearing that? Okay, that's got a favorite now. All right, there's good reason for it. This is before splicing was discovered. They used to say, oh yes, 1 gene codes for 1 protein and that is it. Wrong. Now that we discovered splicing, 1 gene can actually code for a whole family of proteins. Now most of the time they're going to have similar functions, but not all the time. For instance, you can have cases where this might code for different versions of a transcription factor. This one increases transcription. This one increases a little bit. This one inhibits transcription. This one binds to a different enhancer and regulates a different set of genes. All from the same precursor messenger RNA. A little thing I like to mention about this. Another thing I like to mention about this, we'll see how alternate splicing works later on, is a little table here. Let's take a look at a number of different organisms and just see, since we've sequenced so many genomes, let's just see how many genes we find in certain types of organisms. Okay, now if you look at a typical bacteria, okay, typically you're going to have about 2,000 to 3,000 genes. Now there are some of those primitive archaeans have little more than 500 or 600 genes. There's lines of evidence suggesting that to make a minimal functioning cell, you're probably going to need about 500 genes. Different approaches give different numbers. Some say as much as 700 or so. Many of these ones with very small genomes are actually parasites on other types of organisms and stuff. So they don't need certain genes because the host is going to be doing digestion for them or whatever. Okay, so maybe around 500 is a minimal to make a life form. Okay. Oh, God, I can't even spell bacteria. Now, some of the more complex bacteria, like E. coli, the guy that lives in our digestive tract, and the horribly nasty version you get from eating raw hamburger and stuff like that, that actually has about 5,000 genes, and that's quite a bit for a bacteria. So, typical bacteria. Now, the first eukaryote that was ever synthesized was a single-cell fungus, the yeast, and it's got about 6,000 genes. Some of the more complex protists, that includes things like the Plasmodium, that's the protist that causes malaria. Now, what these guys do is, these guys have run through multiple stages of different parts of the life cycle in different hosts. And they constantly change their surface protein so they stay one step ahead of the host's immune system. Okay, now they're fairly complex. They have in the order of about 10,000 genes. Have anybody ever heard of something called a slime mold? I love that name. It just sounds disgusting. Okay, slime mold look, they're well named. If you see them on a forest floor, they may look like kind of clearish, sometimes yellowish or whatever, mass of slime. It's a little small thing. But what they are aggregation of amoeba-like cells. Now what happens with slime mold is many times they're individual amoeba-like cells and they go out and crawl around the forest floor and eat bacteria. And then when they start starving, they run out of bacteria, what they do is they start secreting chemicals that attract each other. So it's like calling a big town hall meeting. Hey guys, we're all starving. Let's get together and figure out what to do about it. So they all aggregate to form the slimy part, okay? And then they start, certain cells develop to form a stalk, or multiple stalks in some cases, and a little spore capsule, and certain cells differentiate into spores. And then what happens, the spores get dispersed, and if they land in a good environment, they germinate. So they have a little bit of development. One time slime mold was the big friend of developmental biologists because it was a simple developmental process. And it's like, what genes are being expressed when these cells turn into stalk cells and into spores and things like that. So a lot of work on that. And not surprising, there was a push to sequence the slime mold genome. They finally did it. So we'll say complex protists tend to be about 10,000 to 12,000 genes. Now, first multicellular animal that had its genome sequenced was this little tiny roundworm that's widely used for developmental biologists and genetics called C. elegans. The genus name is virtually unpronounceable, so everybody just says first letter of C. Okay, it's a little tiny free-living nematode, roundworm. It's about the thickness of a human hair and a millimeter long. It goes through its whole life cycle in 2 weeks. They're very easy to raise in the lab, and there's all kinds of mutant versions around. And not surprisingly, since developmental biologists love these little guys, they sequence the genome. First one, full whopping 250 million bases of DNA. It took years to do that, but we'll say they found this guy had about 18,500 genes in it. Now, another friend of developmental biologists, they also went after its genome, and we find similar things with other insects. People have sequenced numerous insect genomes at this point. Like, tsetse flies, usually the disease vectors. Anopheles, mosquito, one that carries malaria. Tsetse flies are the ones that carry sleeping sickness. That kind of stuff. So anyway, we're looking at this, and let's look at our old developmental biologist friend, the fruit fly. Okay, they count up the genes in that, and what we got here? Uh-oh. Only about 12,500. Whoa, that's substantially less than the nematodes, hardly any more than these complex single-cell protists. And then when we finally go to vertebrates, we'll use the human genome as an example. It's simpler for all vertebrates. About 22,000 genes for humans. Vertebrates seem to have about 20 to 25,000 genes. Likewise, angiosperms, the most complex of the plants, tend to have a similar number of genes. But we look at it, what's wrong with this picture, folks? Well, look at this, and we've got about 20% more genes than this little tiny roundworm. We've got only twice as many genes as a fruit fly. Does that mean we are hardly more complex than that little tiny roundworm of a few thousand cells? Well, if you're in Congress, that may well be the case, but if you're not, it doesn't seem to be the case. So how does a so much larger, more complex organism get by with so few genes compared to what we see over here? Well, this, when we start crossing this boundary from things like relatively simple animals like nematodes and stuff to more complex things, I mean the arthropods, that branch. I don't know if they've done sequenced mollusk DNA, mollusk genomes yet. Maybe they have, but when you start going off that branch, or you go off the chordate branch, that's when you start seeing lots of alternate splicing. You don't see so much in these nematodes. Right? There may be a little bit, but not much. So in other words, every protein they have to make, or almost all the proteins they have to make, they have to have a single individual gene coding for that. For us, if you have a lot of introns and exons, one gene could code for hundreds, and in some cases probably thousands of different cell-specific versions of that protein, just by alternate splicing. So that's a very cool thing. And as we're going to see when we look at alternate splicing, it's not just discarding whole exons and stuff. Sometimes you can discard part of an exon deliberately. Sometimes you might even keep some intron sequences in, deliberately. So not only is it what exons do you keep and what do you toss, it might be what parts of what exons you keep. Or maybe you might even keep a few intron sequences here and there. So splicing gets a lot more, alternate splicing has a lot more complexity than just selecting which exons you keep and which you toss. Okay, so we'll see that when we come back. We'll look at how the spliceosome assembled, the crude thing of the process of splicing, and we'll see you on Tuesday, and then of course, we'll see what goes on lab-wise, by the way. I haven't seen